I will be talking about how we handle research data in biology. So I come from an institute that's mainly dealing with biology and from the Department of Biotechnology and Systems Biology. And I'm working in the working group of called Omics. It's led by Professor Christina Gruden, and the main research topics that we are covering are so shown here on this systems biology cycle. So we start with usually doing some kind of omics analysis, so really untargeted either genomics, transcriptomics, proteomics, or metabolomics study. So we gather a lot of data from quite several samples. So mainly we are studying the responses of plants to biotic and abiotic stressors. And so with these global approaches, we are collecting up to several hundred gigabytes of data per sample. And then we, of course, analyze this data, and we do it bioinformatically, statistically, and some of my colleagues are also doing some modeling. So they're using this data for models. And using that, we are postulating hypotheses, and those hypotheses are then tested in new functional studies. So we either produce GMO plants or do some experiments with treatments. And this then again leads usually to another omics kind of experiments. So this cycle then turns around. So we gather data in basically all steps. Okay, no, it's not. So what kind of data we generate? We generate raw data, then we generate metadata, and also then results, which are either intermediate or final. And in the end, we produce some publications. These are either articles, reports, presentations, posters. So Dr. Kapoor already mentioned this. So metadata is really important. Without that, raw data is basically useless. So you have to know how you produced that raw data, what was sampled, how it was analyzed in the lab, and also how then, when you're producing results, what kind of software you're using, and so on. So also mentioned by Dr. Kapoor, data management plans. So this became very important recently. So also by the funding agencies. So before you start a project, you already have to think about your data that you will generate. And especially if you have bigger projects or multinational different partners, it's very useful. So in our experience, to make an overview table and then say, so who will generate the data, what will be the formats, where will you store it after the raw data when you generate it, and how big it will be. So after you have analyzed the data, where will you store that? So then we have minimal information standards that you can set here already, and also which community standards or data formats you will use, ontologies, and also at the end. So here, where will you store all the data so that it will be transparent and that it will be long lasting. So this is an example from one European project in which we were leading the data management. So after you have your data and analysis done, you usually want to publish, and then the publishers usually require you to submit your raw data to some of the repositories. So in biology and biotechnology, you have some specialized repositories here shown on the left, some of them for nucleotide sequences, then this one is from metabolites, but you can have also like this EDAL is for plant genomics and phenomics. So very different data formats and also metadata standards that you have to oblige when you submit here. And then you have general data repositories such as Zenodo, Figshare, Fairdom Hub, or for computer code, you have GitHub, as already mentioned by previous presenter. So these are both options how you can share the data, and then usually you can also publish it in journal. So this slide is just to show you how quickly the data is growing in high throughput sequencing of DNA and RNA. So you can see this is from 2022. You have 160 trillions of sequences. So and this is from the ENA database, and the SRA database is actually synchronized. So this is, they are copied. One, this is the European one, and this is the from USA. And you can see here the amount of data storage that is needed is, it was in year 21, it was already like 16 petabytes of data. So 1 petabyte is 1,000 terabytes, so it's really a huge database. So just to show you quickly how the position of raw data to this SRA looks like. You have to fill out this metadata kind of, usually you get an Excel table or you have a wizard, depending what you choose. And so you have to have at disposal all these metadata so that you can submit it. So if you just have the raw data, you will not be able to submit. And it's really important then that you do this collecting of metadata already when you are doing your experiments, producing the data. So at the more general repository Zenodo, you don't need to really submit much of metadata. So here is basically a free text what you want to share. And you can have also like closed access here, so you just share the metadata. This one is not yet released to the public. I think this was quite well covered by the previous presenter. So I will just say that fair data is just, as Dr. Kapur said, a very general umbrella of recommendations how you should manage your data. So for biology or biotechnology, I think very important things are here ontologies and gene identifiers. So these are things where sometimes we have problems, especially versions of gene identifiers, and then things are not interoperable. So people in the dry lab have problems. So in the end, it is important for reanalysis that you have metadata with useful attributes, so useful descriptions. Otherwise, you will not be able to do anything with the data. So, of course, after this, what I've shown here is after you already have finished the process of collecting, analyzing, and depositing. So how you manage the data before you upload it to the public repositories. And at our institute, we are managing this so that we have raw data and some really small or really fundamental metadata, so not really rich, stored in a network storage server with backup, and this server has restricted write privileges, so only 2 administrators that can put data on, but everybody can read it at the institute. And so this is, here you should store your data immediately after you collected it, so that if the drive where you have it on the machine, it corrupt, when it's corrupt, you still have the data here backup, and the minimal metadata is just to know what is there, so for easier findability. So then we are analyzing our data either on our computers, or we have a few servers that we named here with so Linux servers for data analysis that is more demanding on the computational side. So here we store intermediate results and then also final results of data analysis and modeling. And we put also then this together with the publications, reports, and other research records into another network storage server that is also backed up. And we have a system developed for managing data, so to organize it. And we developed it at our department. It's called PISA3. So we do not store raw data here because this would, we already have this other server. We just have links to this server so that, or to public repositories in the metadata so that we know where the data is stored. So this PISA3 system was developed basically for our own need to organize the projects, because we work on many projects as a team. And before that system, everybody had his own organization of where he put his data, directories. Some people were collecting metadata, some were not. So this is kind of standardizing this on our first in our group. So we developed batch scripts. So in Windows, they work in Windows operating system, and these scripts generate directory tree structure based on this ISA framework. The ISA means these levels here: investigation, study, essay. But we also added project level since we are all working on projects basically. And these 4 levels are somehow enough for us to store all the data and metadata so it covers our needs. And the metadata files, we have decided that they will be in a tab delimited format, and that they will be really collected on the fly. So when you are executing this batch script, you are already putting in the metadata, or your directory tree will not be produced. So this system, you can download it from GitHub, and we also have an article preprint on bioRxiv, and soon we hope it will be also published in a journal. The creation of these levels and simultaneously collecting of this metadata is done in such running these scripts that where you actually sometimes, like, the title is just free. You just put your text in. It's free text. If you don't put anything, there will be an asterisk showing that you should put later something here. So you don't need to put everything at this creation step. But some of these are just selecting from the options. So here you select which homogenization protocol that is already predefined in a template you are using. And that way, they're trying to make it easier for researchers to really collect this metadata. So in the Windows Explorer, this is for one project. Here you can see how this structure looks like. I'm highlighting here this PISA, basically ISA structure. And you can see we show, we basically automatically name the levels here with at the beginning, so that you know where you are, in which level you are. And this last level, the essay level, you have specific, so based on different essays, have specific directories where you put your. Here there is no input because you're isolating the RNA. You don't have any files as an input, but you have outputs and reports. But in some dry lab essays, you will have inputs and also like scripts directories. So this is how it's automatically organizing the structure so that everybody is basically synchronized in our department. This is just an example of metadata at the essay stage. You can see how what we are measuring, what kind of metadata we are collecting for RNA isolation, and so this metadata can easily be then opened in text editor, and they can be edited. But what is crucial is that it's a tab separated file. So also with some computer language, you can read it so into some computer pipelines. So, this is the kind of a showcase of interoperability of PISA-3. So we also developed two R packages. So R is a programming language for statisticians. And so the PISAR package is basically used to read the structure and metadata from these metadata files. And also it can produce output, so exports in ISATAP metadata standard. And you can also zip, of course, the whole directory structure. So these two files then are can be directly uploaded to Zenodo or Figshare or other general data repositories. But we also for upload to the repository Ferdom Hub, we created another package, SIG-R, which is using the SIG API to upload the whole directory structure to Ferdom Hub. And basically then you have all the files in this public repository. The good thing about Ferdom Hub is that it uses the same kind of ISA structure and the ISATAP format. and it offers many open licenses for data and code, and also it can produce DOIs for your data. So that's why we decided first to go to develop upload for this repository. And this is, you can see the same essay here on our network server. And then this structure basically is copied to Ferdom Hub, and you have all the files also here available. And you can make it open, and at that, then the research, your research is really transparent, and people can access it. So just at the end, I would like to make to show that there is also one interesting online guide for data management practices for the entire life cycle. So this is it's called RDM Kit or Research Data Management Kit from Elixir project Converge, and you can see here this is the currently what domains they are covering. It's aimed at both researchers and data managers, also policymakers, and they have really good pages not only for certain disciplines, but also general ones such as how to do the data management plan, which might be interesting for PhD students also, because there are some templates, and basically they're showing considerations and also solutions for this. And at the end, just here to acknowledge the group. So people that were working on PyZ3 development. So Christina, the leader, and here you have my email also if you need, if you want to ask questions or if you want to contact later.